Yeah, good morning, everyone. First of all, actually, I would like to get an impression of who you are, who I'm talking to, so I communicate on the right level. So who of you is working in AI right now? Most of you. Who has a computer science or data science background in terms of studies? So also most. OK, that's good. That's what I kind of expected. Yeah, I'm also a trained computer scientist, and I actually also studied at Imperial College for a year abroad. And I remember back in the days, so I started studying in 1999. Basically, everybody told me that AI is never going to work. So after finishing my studies, I went up to my future PhD advisor, and I said, OK, I would like to apply AI in medical imaging. And he said it's the most stupid idea he has ever heard. But as you all know, things have changed. This is a list of the 521st products that were certified by the FDA. And what you can see from it is, that the leading applications are actually in imaging, radiology. So we distinguish software in a medical device, which is something that you would use, for example, to help you reconstruct an image, or software as a medical device, which would be, for example, a deep learning application to diagnose a certain disease. So that's great. So imaging is one of the key applications at the moment. But something that we have to keep in mind, I think, is that deployment or certification is not equal to benefit. And there have been prominent examples. I'm sure you've seen some of them. For example, that was a Google product that performed great in research papers, great performance and everything. And then they rolled it out in Thailand in a number of hospitals. And it turned out that it didn't work as well as hoped for. So, I mean, the authors formulated it quite cautiously. But in the end, what happened, 20% of the cases of the patients that came, this was for detection of a diabetic eye disease, the system didn't do anything because basically the data was out of distribution. And in those cases for which it worked, the authors didn't even report the performance. They just said there was a radical drop. So apparently it wasn't that good. And there's many other examples. This one, for example, was for sepsis detection. You're probably all familiar with AUROCs, AUC curves, where basically we plot the sensitivity, the fraction of positives against basically fraction of negatives detected. Then you get this curve, you compute the area under it, and that gives you a performance measure. So, and the advertised value was, for example, much, much worse compared to what was found by external validation, which is, of course, critical. And it actually is clinically quite important because in these cases, 2/3 of the sepsis cases were actually missed. So you see, I mean, even if we deploy systems, even if we make it all the way to translation, it still doesn't mean that we get all the benefit that we see in research. So of course, I'm primarily a researcher. This is an analysis that we did, published this year at CVPR, where we looked at all the imaging competitions that take place at the major conferences. And we wanted to shed light on whether these competitions actually generate scientific progress. I mean, if you work in data science, I'm sure you know all these competitions like ImageNet, for example. I see some nodding. So this is really how you benchmark algorithms these days, right? So we wanted to shed some light on it. I'm not going to talk in detail about this paper, but something that I found interesting is that when we asked the organizers, we analyzed 80 competitions. If the problem is solved, then basically the majority of problems was not yet solved. So in a way, that's also a good thing because it means that in the imaging domain, we still have some work to do. So let's look a little bit into why problems are not yet solved or why they don't work when we deploy them. Have you seen this paper? Has anybody seen this paper before? I really enjoyed reading it when it came out because it really reflected my perception of the things that are going wrong. So this was about COVID-19 detection, and you see here on the x-axis a lot of publications on the topic, and the colored bars are basically data sets that were used for this specific question. And of course, all the papers claimed that now COVID detection is possible. But if you look closer, then you will see that a lot of the groups used the yellow and the blue data set. And then if you look even closer, you see that the control data group, the no COVID, comprised only infants, and the COVID-19 group comprised only adults. I mean, this is ridiculous, right? It went to this extent that sometimes only these data sets were used, so meaning that all that an algorithm has to do is actually to distinguish children from adults, right? And that is a problem, and you could argue, but, well, that's trivial. I mean, age as the founder has been known for many years, but you see, it did happen at scale. So I think we do have a problem of people rushing into the topic, not really knowing what they're doing, and then publishing some fancy results. So I want to talk a little bit about this. That's why I called my talk "Question Everything." So I think, I guess the German industry has been popular for the cars. I think in terms of AI, they're not doing that good. But I'm showing it because I think if Germany represented something here, then it was quality. And I think the reason was that they optimized every single component of their product, of their production pipeline. And I think that we have to do actually the same in imaging. And from my point of view, we put too much effort into just the image analysis. Of course, I mean, I'm a computer scientist. I also want to solve problems, and this is the most exciting part. But it's not the only thing that matters. There's much more around it, and that's actually my point. So this is my, let's say, perspective that I can share with you. I already told you I'm a trained computer scientist. I'm also one of the fellows representing the topic of AI and health in the framework ALICE. Who has heard of ALICE before? Yeah, okay. Well, fewer people than I hoped for. So hopefully it's going to be a really well-known AI initiative in Europe in the future. Well, I've also been editor of Nature Scientific Data, so I have a sort of data view, and I'm one of the directors of the National Center for Tumor Diseases, so this gives me the health perspective. My main domain, but I will make this broader than this, is actually surgery. Why surgery? If you look here, death within 30 days after surgery has actually been identified as one of the leading causes of death worldwide. And I believe that we're looking far too little into this topic. I mean, of course, other domains are also exciting, and specifically radiology is good because we have a lot of data. I mean, and from a process point of view, it's easy because you have the images, you only need to annotate them, you can already do something. But I think in terms of gain, there's a lot here to gain in surgery. So basically, what we want to get away from, the experts in their subjective decision-making to a world in which we interconnect all the information and improve surgical safety. And I've been working on this with a great team of clinicians from several hospitals, also with technical collaborators. And then, of course, my team. This is the most recent group picture that we took, so many thanks to all the contributors. Okay, so I will start with something maybe unusual, which is shown as the last step here. And that's for a reason. I'm sure you've followed or at least seen the AlphaFold success story. Yeah, now I see a lot of nodding. DeepMind solving protein folding. And one of the core team members, Simon Kohl, was a former colleague of mine. And I asked him, so what do you think was the key to success of this project? And he said it was fixing the validation strategy at the very beginning. So they spent a lot of time thinking about how they will actually benchmark success. To me, I mean, I've been advocating this for a long time. This was like, I don't know the English phrasing, but it was just honey in my brain, basically, that somebody who had such a success story to show actually made that statement. So I want to give you a few examples why I think this is so important. I don't know if you know the Cobra effect, or if you've heard this story. Yes, I see some nodding as well. So, well, there was a rat plague in Hanoi, and the government thought about how can we get rid of the rats. So they said, okay, you get a monetary award for all the for a rat tail that you hand in. So they use this tail as a proxy for killing a rat, basically. In practice, of course, what did people do? They cut off the tails, left the rats alive, because they knew they would reproduce and then they could earn more money, right? So it's a sad story, but it's a good story also for an example of a metric that fails to reflect what we actually want to measure. And I think we have many of such examples in AI as well. So the practical problem is that what we have is, I mean, how do we measure success in terms of metrics? So let's say you have a reference, radiology, annotation. Imagine these as tumors, for example. And then you have two predictions. Now, the job is basically to decide which algorithm performs better, or how do you measure it? So already quite a few years back, 2017, 18, we looked into this and we found this is the metrics that you see are most commonly applied in competitions, imaging competitions. And I'm sure you've heard of the dissimilarity coefficient or the IOU. They're almost the same mathematically. So what they do is basically they look at the overlap and they look at the individual surfaces basically, and then you compute a value that is between 0, no overlap, and 1, full overlap. I mean, you see the formula here. So I would say, at first sight, this would make sense, right? Now, let's look at an example. So this is an algorithm that, according to common validation standards, so the DICE is used in the international competitions. It's used by hundreds of labs worldwide. This is an algorithm that, according to that validation, could replace a radiologist. So if we would take this algorithm, so again, you see the output of the algorithm here, the reference here, this would be a disaster clinically for the patients. Why? Because now I'm scrolling through the data set, I'm taking another slice. You see there's a small lesion annotated by the radiologist, missed by the algorithm. So how is it possible that this algorithm was assessed so well? It's because we're basically counting pixels rather than objects, right? So imagine this is we're talking about cubicle here. We have 10,000s of pixels in the big tumor. We have just a few pixels there. That's basically noise in the validation. And the same we do in training. So the algorithm with this specific metric is not trained to detect all the lesions, which is what matters clinically. So same here. I mean, now schematically shown. The DICE, for example, would favor this algorithm over that one, although this one is doing what we actually want much more, right? Detecting all the objects. And I mean, of course, in this case, the solution is to phrase the problem differently. We need it to be an object detection problem and not a segmentation problem. So there's a flaw already in the phrasing of the problem. And it's not, you know, a corner case. This is happening all the time. And I just talked also to some clinicians. They wanted my advice on a challenge. They wanted to do the same thing. And I said, okay, but this doesn't make sense because don't you want to detect all the lesions? And they said, yes. I said, yeah, okay, so then you have to phrase it as a detection problem. And he said, no, but we cannot annotate it in this way. This is too complicated. We can just annotate one color basically because we don't know exactly sometimes the boundaries, so we have to phrase it as a segmentation problem. I wasn't very happy about this answer. But you see there's a lot of practical problems sometimes. Second example, so here, this is a project that we have. I mean, maybe you've seen the success stories in polyp detection. I think that's actually some of the interventional AI success stories that we can now detect polyps quite well. We now have a project on capsule endoscopy, meaning that you swallow a capsule, and then it's very cumbersome for the clinicians to go through hours of video material to detect a tumor, and we want to do it with machine learning. It's not as trivial, so the images are actually quite hard to interpret. Anyway, let's talk about the metrics. One decision that you have to make, because typically you will see a box of your detection algorithm, right? And then you have some reference annotation. For example, also a box could also be a mask. And then you have to decide what is a hit, what is a true positive, right? That's a decision that needs to be made. And this decision can be made pretty wrong as well. So I show you an example. Here, let's say we have output of the algorithms, left and right again. You see the first algorithm actually doesn't detect, when you look at the reference, doesn't detect 2 out of the 4 tumors that we have. This is just a schematic example, but of course we analyze this quantitatively properly, but just for illustration. And now if you take a common metric, you will see that according to average precision, which is the common metric, configured with a common threshold for detection, you would actually get the same performance. And that's, of course, a little bit disturbing. So in this particular case, what we did is we asked multiple clinicians, what do you actually think is a hit? And it turned out it was completely different from the way that the machine learning community configures the metric. So is somebody working in who's working in object detection? A few, yeah. So you will know that the COCO, for example, they basically take the threshold 0.5 to 0.95, which is quite strict. And it turned out the clinicians wanted more something like 0.2. So again, we have the situation that we're measuring something that doesn't reflect what we need. So this means our whole research might be going in exactly the wrong direction. Third example, prevalences. Also, I think maybe underestimated topic. So who of you is working in classification? Who has worked with classification metrics before? So quite a few, that's good. So let's see if I can tell you something new. So obviously, I mean, you're aware of the problem of prevalences in algorithm training, because this is where it's discussed a lot, I think. But it's a very realistic setting. We've all seen it in COVID, right? I had a hard time explaining to my family that if they have a positive COVID test, the likelihood that they actually have COVID depends on the prevalence. It's very not intuitive, I think. So I had a very hard time here. But let's look at what prevalence shifts mean in practice. So I've shown you before that accuracy and F1 score, and I'm sure you would agree with me, are some of the most widely used metrics. So let's now take a test or an algorithm that has an inherent sensitivity and specificity. That's really the properties that matter, right? Now, if we change the prevalence, we can really have a radical shift in the metrics, although we didn't even change the algorithm. And I mean, here we talk about a delta and F1 score of 0.7. This was cherry-picked, but I've come to a more practical, reasonable example now, but it should illustrate the effect. We have a problem here. And I think we also have a problem because, especially in medicine, I don't know in which domain you're working and if it's the same problem, but in medicine, we tend to say, well, we achieved this accuracy of 1 score and others achieved that. I mean, it doesn't make sense, right? So let's look at it a little bit more systematically. So here we have 30 different classification tasks. Some binary, some not, with different imbalance ratios in the data. And for now, for simplicity, we assume that the deployment prevalences are known approximately. We've done robustness analysis, and there's ways to also estimate prevalences from the data, et cetera, but I will not present all the results, but for simplicity, let's assume that. All right, so now what we look at, so the colors are a bit weird, but I think you can still see it. So you will soon see a plot here. Well, basically, we will look at the difference in to the metric score on the deployment test set. So this is how we trained. So we trained with balanced training, which is something that a lot of people do. And our test set during development phase was also balanced. And now we just artificially basically change the prevalence on our deployment data set, which otherwise is totally in distribution. There's no other distribution shift. It's just the prevalences that we change. So what happens? A lot happens. So we get all of a sudden a totally different result. And that's, again, a huge difference, right? So this is just to illustrate that my COVID example wasn't totally off. So we learned from this that popular metrics are not really comparable if you change prevalences. Now the question, who has heard of expected cost as a metric? So not so many. This is interesting because I also wasn't aware of it for a long time. So I can also tease you, I will come to this in a second, that we did, we joined a huge international consortium to talk about metrics and best practices and all that. And we put our first draft of recommendations online on social media and asked for community feedback, and also people were invited to self-nominate themselves for joining the initiative. And then there was a speech recognition expert from Argentina. So she had never worked in healthcare, never worked in imaging, but she said, yeah, but I think you're missing a very important metric, which is expected cost in your recommendations. And we looked into it, and we finally actually agreed with her. I found this very interesting, but more on this later. So what is expected cost? So you all know what accuracy is. Accuracy is basically a fraction of correctly classified samples divided by all the samples. Simple. And it's actually 1 minus error rate. Now, if you phrase this a little bit differently, the error rate, which you can basically say it's identical to accuracy, is 1 minus, you can formulate the error rate as a function of prevalences. Prevalences, so again, meaning the frequencies of the classes, positive class, negative class. And then the R's are basically the fraction of misclassified samples for that class. So it's just a reformulation of the same thing. Now, expected cost is really just a generalization of this, of the error rate, but you add a cost factor, which makes a lot of sense because you may say that certain class confusions, confusing positive with negative or vice versa, or same in multi-class cases, are more severe than others, right? So for example, when you have a classification problem where one class is no tumor and other classes are different stages of tumor, you would agree with me that it's okay-ish to mix up two tumor classes, but not the positive and negative, right? So this is would be an ordinal classification problem where you want to give more weight to certain class confusions. So this is what you can use the cost factor for. Now, if you use 0-1 costs, meaning 0 costs if you have the right class, 1 for all others, you again end up basically with the error rate. Why am I telling you this? Because we thought this is a great opportunity because now we can just replace the prevalence that we have, the formula, by the prevalence that we expect to have upon deployment, right? And there's nothing else you need for it. And then if you do this, you see that you actually have a stable result. So that metric basically enables you to compare results across data sets. Okay, so I will come back to this. But before this, I was very glad about these statements by, let's say, well-known also companies or leading AI companies. We had a Google Health person, Sheikh Azizi here, talking about large language models recently in our data science seminar. And when she said, what's missing, I was very excited to see 2 points that were related to validation benchmarking. Very interesting, I think. And then also OpenAI said, okay, this is really an overlooked topic. How do we measure success? Yeah, and this is why we found it. Yeah, you must have recognized the matrix analogy here. We founded Metrics Reloaded, where basically based on an initial consortium, we gradually invited people to join, or they self-nominated themselves. I gave you Luciana as an example, the Argentinian. And we wanted to revisit metrics, that's Metrics Reloaded, to see if we can make progress on that end. So many thanks, by the way, to Annika and Paul. We have been working on this very, very intensely, actually, as the core team. Okay, so the first task was to come up with a scope. I mean, validation is very broad. You cannot solve everything, but we decided to be basically independent of modality and domain, and look at classification at various scales. So basically, semantic segmentation would be pixel level classification, image level classification is clear, and then object detection is something like object level classification, right? And then you have instance segmentation where you not only classify each pixel, but you also distinguish instances, which is what we really would have needed in the brain tumor example, right? Okay, so what did we do? How did we approach the problem? We thought, okay, we want to be independent of domains, so let's think about what really matters to select the right metric. So we abstracted from the modality, and the important thing is, I mean, do you have small structures relative to the grid size? Do you have overlapping structures? Is there size variability? Do you have a class imbalance? What are you actually interested in? Does the boundary really matter or not? Do you need the center to be correct? That's in cell tracking, this can be important, for example. Does the volume have to be correct? And so on. So we try to extract what actually matters. So I can maybe show you. So that's now a tool that we developed to make people's lives a bit easier because the recommendations are quite complex. So there's a few functions. This is the one on the recommendation, so you can basically choose your topic. And then step by step, you're guided through this process. So here you, for example, asked, is the size of the structures small or large with respect to grid size? Are you interested in the center point? You can read what we mean by it. Do you have tubular structures? And so on. And then step by step, you get the metrics, the complementary metrics that you want to have for your project problem. And of course, this is not perfect. It can never be perfect, I think, but I think it gives you a starting point. And if you're interested, maybe something useful also that we have on the website is the metric profile. So it gives you, basically at a glance, the definitions and the relevant pitfalls and also recommendations on how to use the metrics. That's all online. All right. So I think I made that point probably clear. That really the validation strategy has to be aligned with your problem. Now, let's look at other components of the pipeline. This is something that I wasn't really aware of, or at least not of the effect, and that surprised me a lot. So when we talked about metrics, it became clear that we have to distinguish prevalence-dependent from prevalence-independent metrics, because that's a very relevant differentiation. So when we started thinking about prevalence more and more, I realized how relevant they actually are, not only when you train your algorithms. So let's look into it. First of all, as a motivation, this is a system, Olympus polyp detection, that was actually very successful. Here you see the results in the UK. That's basically internal validation, very good sensitivity. But somebody found that when they applied it on different data, the performance dropped. Now, of course, it's very hard to tell why this happened, right? And in my group, we look into a lot of domain shifts that you may have. So geometrical, for example, in surgery, if you start manipulating the data, the geometry will look very different. You may want to compensate for this. Sim to real, a label shift. But let's look into a prevalence shift today. So most of you are probably familiar with this picture. So you have a neural network, some logits. In the end, you apply softmax, you have some output. Now, there's one important decision that sometimes we oversee, which is how do we actually convert that softmax output into something that can be handled by a confusion matrix? So if you ask this to a junior student, they will not even understand the question. They will say, well, it's obvious, I take the biggest value. I mean, what else should I do? And mathematically speaking, that means you use the argmax operator, right? And again, with the juniors, I think they don't even understand what else you should do if you have such an output. Okay, so what's the problem? Again, same setting, 30 different classification tasks, so we actually can draw broad conclusions from my point of view. Now we will look at the delta from what you can do best if you choose an optimal threshold. In this case, I think, no. Imagine it to be binary tasks. It's not, but just for simplicity, it's good enough to imagine it to be binary tasks where you actually set your threshold between 0 and 1 for positive and negative, right? So the argmax would mean 0.5, right? So now we look at the delta between what you get with argmax to what you could get if you optimized your threshold. And again, you see a large deviation. And this is, I think, very important. Because if you think about what it means you train an algorithm in this setting, balanced training, which a lot of people do. Then you deploy it with a, I don't know, 1% prevalence, which is totally reasonable for many diseases. And you don't get a good result. This is not a good result. I mean, take any sample here. A difference in accuracy between 0.8 and 0.9 or 0.86 and 0.92 is very relevant, right? I mean, if you think about the machine learning publications, you know, the bold-faced numbers in the tables, what differences are we talking about? This is a game changer. Okay, so what's happening? Does anybody have a proposal why this is the case? Well, this is the development data. We had 50-50. We optimized. We had the optimal cut, right? Now we changed the prevalence. This means we are in a position like this. This means the 0.5 doesn't make sense. We have to shift the threshold to have the best possible accuracy in this case we're talking about. And the core problem really is that we didn't recalibrate the algorithm. Okay, so from a theory perspective, simple Bayesian framework, if you look at expected cost, this is the formula for getting the best possible threshold if you want to optimize for accuracy. If you simplify to accuracy, sorry, if you optimize for expected cost, for simple accuracy, it's actually the argmax operator. But that only holds true if the class scores are calibrated. This is very, very important. So what does calibration mean? I'm not claiming expected calibration error to be a very good metric. You can read all the details in the paper, but I think it's very good for illustration. So what do you want? You want your output scores, the softmax scores, to reflect the confidence, right? So if you have an output of 0.8, so then that means you want 0.8 of the cases in which you have that value, 0.8, to be correct, right? So the empirical accuracy should be 0.8 if you have a confidence of 0.8. So to measure this, you actually put, or this is one way to do it, binning is biased, but again, side story. So you can put all the prediction with a certain confidence in the respective bin, and then you compute the empirical accuracy, and then that basically should be lie on the diagonal. Then your system is calibrated. Okay, now I will not go into all the details of what you can do also in the calibration part. If you're interested, you should read the paper, our Mika paper here. But end of the story is that if you recalibrate using the deployment prevalences again, and temperature scaling would not do the job because it doesn't contain a bias term, then you're actually fine. So again, much more details in the paper. I don't want to bore you, but I think for me this was really interesting because of the many tasks that we validated, and we actually saw that we have an actual problem and not just a minor problem. All right, now beginning of the pipeline. So sorry, I will have not too many, but a few bloody images because that's the kind of data that I work with. But I think the message comes across well because you see that it's very, very hard to distinguish anything from anything, right? And that's what a surgeon sees when operating you. So they don't see, of course, what is underneath the surface, but also they cannot really distinguish different structures very well. And from my point of view, and now we come back to the question everything, a problem is that we're trying to imitate the human eye. I mean, you know, we are sensitive to red, green, and blue. This is how our eyes are built. And this is how the chip industry also built the cameras, right? They use RGB chips. I mean, you know, you're working with RGB images, right? So I think it doesn't make sense, and we should do something different. So just imagine we would not capture an RGB image, but we would actually capture many images corresponding to different frequencies of light. This is basically then gives you in each pixel a whole value of measurements, which we call a spectrum. So why would we do this? We would do it because if you look at the molecules here, I mean, you're probably more data scientists, you're not maybe familiar with all of it, well, this one you know. But hemoglobin is basically blood, a main constituent in blood, and it can be bound to oxygen or not. And I'm sure you know that the oxygenation in tissue is very important, right? This is what you measure, at least systemically, with these clips in the intensive care unit, right? So that oxygenation is important. Here we have the two main components for assessing oxygenation. We have fat, water, and you see that all these different molecules absorb light at different frequencies really differently, right? And the hypothesis was that this can actually help us recover the tissue composition. And I will, because I know you're data scientists, I will not bore you with the details, but I want to show you the kind of things that we're doing. So for example, here you see, and this is basically all done with AI. So we basically use the deep learning algorithm to recover from the images that are not human interpretable the values that we're interested in. So here you see an example of kidney transplantation where you see there's a part of the tissue that is not very well perfused, right? So we can actually recover oxygenation in a spatially resolved manner. We use it to optimize surgical techniques. So in this case, anastomosis is basically something to sew two structures together again. For example, imagine removing a tumor and then you need to, let's say in the colon, and then you have to put the tissue together again, and then it's very relevant that it's well perfused. But so far, we didn't have a method to actually look at perfusion in a spatially resolved manner. I mean, the clips don't help us there, right? So now we can look at different techniques of doing, for example, an anastomosis. We can also measure effects of medication that we give intraoperatively, because now we can see perfusion. This was a fancy application where a neurosurgeon saw our kidney applications and he said, yeah, I really need this method to measure oxygenation in the brain. And he explained to me that when you have a stroke or also a migraine, there's this phenomenon called spreading depolarization in the brain. This is a fast-forwarded movie. Normally it lasts a couple of minutes, where basically the oxygen consumption in the brain changes in waves. And he says he needs to be able to measure those such that he can develop better medication against stroke. And then, sepsis, actually a different application or a different molecule we're measuring. So here the hypothesis was that the microcirculation disorders would show in a change in water concentration actually in the tissue. And now after the first about 200 patients, we think that we can actually measure or diagnose sepsis based on such data. So this is just to pitch you, let's say, the range of possibilities. So when we started to work on it, one of the things that we looked into, and we published way too late, but it was still novel enough, was, do we actually have a benefit of the spectral images when doing simple classification? So for example, semantic scene segmentation. And the answer was yes. But now, today I wanted to talk about problems, about questioning everything. So I want to show you an example of something that happened before we had the new 200 patients. So this was back in 2019-20, when our collaborators had the hypothesis that we could maybe measure sepsis. And here we would need, basically this is the visible range of light, this is near infrared, and the hypothesis was that here, where water starts being absorbed very heavily, we can basically use that data to detect sepsis. So they gave us the data, and my PhD student gave me that plot. And I said, OK, great, let's go to Nature. And I was kidding. I knew this couldn't be right, because that's now the 3 groups: sepsis, healthy volunteers, and another control group of surgery patients, basically. And I was absolutely certain that if you have such a new hypothesis, you cannot have 97% accuracy. It really doesn't make sense. So we looked into the data, and there was something that had bothered me for a while. So this is now spectra corresponding to different organs. And I was confused, or we were all confused, by the fact that we had such a high standard deviation in a certain range of the spectrum. It didn't really make sense. And if there's something that I learned, is not to ignore such observations, because normally they will show you something. And then we realized, and I'm really credit to my team that they found this, because I would have never imagined. They split the data at different time points, and then they found if you do that, you can really get completely different spectra for the same organs. So what happened? We found out after we saw this that the provider of one of our devices that we needed for the measurements had come to the hospital and recalibrated something without us knowing it, and that basically caused a change. And can you imagine how irritating that is? So why was it such a big problem? It was such a big problem because when we then plotted our groups, we saw they were acquired sequentially. And I just wanted to shoot myself, because the data looked so good, and there was no way we could use it, actually. So this was back 2020. Then, by that time, regulation in Germany had changed. It took us a lot of effort to go through new ethics. And now, end of 23, we have the data again. And yeah, my poor PhD student is doing her PhD a little bit longer than 3 years, because she had to wait for the data. So, so that was irritating, but this is what happens. And message being, yeah, I think we really have to take serious if we notice something that doesn't look right, then let's dig into it, because normally there's something behind. OK, now back to this example. So we made that video and made that data analysis, and the surgeon was happy. But then, of course, just analyzing the surface of the brain is not sufficient, right? The brain is 3D. And everything that I showed you so far was surface imaging. And if you think of tumors also, I mean, if we're not talking about, for example, colon, they are typically located underneath the surface. Who has heard of photoacoustic imaging? I see 2 hands. OK, so what is photoacoustic imaging? So this is my institution's logo. We put this sponge thingy and the logo in a milk bath. What would you get if you went to a gynecologist and acquired a 3D image? Well, you would see something like this, right? You can 3D reconstruct the sponge and the sign, but you wouldn't see, or you don't see, I mean, this is an actual reconstruction, what is written on the sign, because ultrasound is not sensitive to optical properties, right? So you can also not see the color of an embryo's skin or the color of an eye, the eyes. So now imagine illuminating the tissue with laser. You illuminate the tissue, and what happens? Again, we can have different frequencies of light here, and we already saw that the tissue will absorb the light as a function, basically, of the molecules that are present, right? So the light is absorbed, the tissue heats up. Because of the heating, there's an expansion, and the expansion leads to an emission of an ultrasound signal, which we can measure. And it's actually working, because so when we started working on this in my ESC grant, this was 10 years ago, there was no device you could buy. So we actually built it ourselves, basically. And I was so surprised when it actually worked. So we would actually light in, sound out. We would measure that light response with our ultrasound device. It was very exciting for me. So what does it mean to our image? It means that we can actually recover the optical properties. And that gives us a very powerful complementary information, right? So again, with our device that we built, which is definitely not perfect, you see the images are much more noisy compared to what we have in hyperspectral imaging. But you also see we have a region of interest here that we can actually monitor the spreading depolarization based on that device. And meanwhile, there's actually a certified device available now for maybe 2 years. And we have access to the data from 3 different locations. So why I'm showing you this, because there's several parameters in the device that unfortunately feature high variability. So for example, the laser pulse energy you see corresponding to the different devices, it changes. Why is this relevant? I'm sure you can guess, because now the neural network will be very sensitive to which device was used to acquire the data, right? And we played around with biased data sets, meaning that we, in a binary use case, actually it's for peripheral artery disease, we basically took more of the positive samples from a certain device and then fewer from the other device. And we see then when training a classifier, this is how the setup basically looks, the schematics. We see in the bias data set, the algorithm concentrates on this area here, which is not where the anatomy actually is, but this is where you see the device properties the best, the artifacts resulting from devices. And only in the unbiased data set, the algorithm actually learns the manifestation of the disease. And I personally think that this is a very, very relevant topic. There's so many things that can go wrong. I mean, especially when you talk about diseases in radiology, the diseased patients will often have cables, right? Or there can be skin markings. Again, we know already that neural networks can classify even the person who acquired the MRI. They can basically detect anything. And whenever this is the case, we have a potential problem in classification, right? Because we have to make sure we don't have a biased data set. Okay. Next step is the image formation. So I already hinted at it when I pitched to you what we can do with these methods. So now we don't want to just take the data and classify. I mean, from a computer science perspective, that's, I guess, trivial. We actually want to recover the content. And I will not go into the simulation details, but what we did, again, this started 10 years back, was, and now it's quite commonly used. Back then, I think that was one of the reasons why I got my ERC starting grant, because we pitched that idea of learning actually from simulations. So we use all our prior knowledge on how light interacts with tissue, on how tissue is composed, on how light interacts with cameras, same with sound. Then we generate synthetic data. So in the most simple case, it's just a spectrum and the corresponding oxygenation, but actually we do more. It's just for illustration. And this was the very first patient that we actually imaged, where we clamp a vessel and release it again, and you see that the oxygenation actually, or the organ is rapidly reperfused again. So this was basically how it started. But again, today I want to talk about the challenges and about the questioning. So what's here to question? Well, in the simple case, this is our setup, right? We have a measurement, a spectrum. Let's, for simplicity, assume we have this pixel-wise. And we get something out, a vector of tissue parameters, including oxygenation. For now, you can even assume we have just one value. So why is that a problem? Well, in theory, we could have, in a tissue, 2 completely different values of oxygenation and taken everything together, tissue depth, layer depth, everything that we have, maybe we would get a similar measurement. Now, having a point estimate as an output may not be the ideal way to go here, right? So we argue, rather than going for a point estimate, a single value, we should actually go for a full posterior in which multiple plausible modes in the output data can be captured. So this is something that we do with invertible neural networks. So I don't have time to go into them. I'm sure many of you heard of normalizing flows before. Exactly, so this is a specific architecture that leverages that concept, and the core idea is that you get not a point estimate, but a posterior out, which can be very useful. So I can show you how it looks in practice here. We look at the very same physical point now. And for that same physical point, our problem can be well-posed, single mode, or ill-posed, depending on where we place the probe. And that's helpful, right? Because then that means I have some, if my problem is ill-posed, I can actually potentially do something against it, which, yeah, is illustrated here. And I think the ambiguity is omnipresent in medicine. I can show you this example here. So here we want to do 2D, 3D registration. So we have a 3D image acquired at some point, which contains a lot of information on the patient, and then we have only a 2D image just before the surgery, and we want to basically estimate where in the 3D world coordinate system our X-ray device was positioned such that we can fuse the information. And again, I mean, a simple case of prone versus supine position, right? You can actually get pretty similar images, meaning that also your solution can be ambiguous. And this, we have shown, can be captured via the multiple modes in the posterior. Okay, let's move to the next point, the last point to cover for me, which is the data management. First of all, labeling instruction. How is imaging data labeled? We asked in our MICAI society, that's the most important medical imaging society, where when you apply for a challenge, you have to fill out a comprehensive form, and you also have to provide the link to the labeling instructions. But it seems like people don't actually have a labeling instruction. So why is this a problem? Let's look at a few examples. So let's say you want to segment instruments in laparoscopic video data. Then, I mean, I've heard this from people. They say, okay, this is trivial, right? You just have to basically outline the metal in the data. But then, as always, the devil is in the detail, right? So for example, here, I mean, when you decide you only annotate the foreground objects, what do you do here? I mean, you see the instrument, right? Should it still not be annotated? It's a transparent trocar. Or now you have an overlay. You could say, yeah, okay, we just ignore it, we cut it out. But what if you have an overview of individual letters? I don't think we want to cut them out one by one. So as you see, again, devil is in the details. And this holds true for many applications. So here, for example, this is a lung tumor, and 10 different radiologists were asked to annotate it. And you can see that they absolutely disagree on the exact outline, right? So, and sometimes this has to do with the instruction. So here, for example, liver segmentation. We have a problem here because there's a large vessel that starts being inside the liver, but then at some point it's outside the liver. So how do you annotate it? Do you always ignore it, or do you choose a certain point spatially when you stop having it in the annotation? So it's things that need to be basically discussed. And when we asked annotators of professional companies, they all agreed that labeling instructions are absolutely crucial, and that more time should be invested in them. Now, what I found interesting in the study that we published this year was the following. So we had 3 types of labeling instructions in the study. One was short text, one was extended text, and one was extended text with images. And our hypothesis was that in each step you will have a radical increase in accuracy when you give this to professional laborers. And this hypothesis was actually wrong. So we didn't get a gain, really, of putting more text. It didn't give a benefit, whereas the pictures really made a I mean, this was basically hypothesis confirmed, this green one here, really made a big difference. Maybe a question to you, have you ever used the service of an annotation company like Amazon Mechanical Turk or something else? Who has? Yeah, a few. So a second question that we had was, how does MTurk compare to these professional annotation companies? So I covered it to separate this inside. And that was a very clear result, actually. Although we did a lot of quality control, Amazon Mechanical Turk really performed much more poorly compared to all the 4 companies that we investigated. All right, so take-home message, solve the right problem, design your validation accordingly, and then also please challenge everything that you do, because hopefully I could show you that it matters. And finally, what's next? I can only show my perspective for medical applications, but I'm sure you're also keen on all the foundation models that are coming out. And there's a nice paper that came out in Nature on foundation models for generalist medical artificial intelligence. I think it's worth reading. It reflects what I think will happen in medical imaging or in healthcare in general. And as you can imagine, it's beyond what we call artificial narrow intelligence, where you have an algorithm trained for a specific task, but instead you go for systems that actually can solve a lot of different tasks, like you see basically in these images here. By the way, who of you is working with ChatGPT or is using ChatGPT quite regularly? I would say half. Me too. So ChatGPT, for example, chose the title for this talk. And do you also use it for, who uses it for coding? Also quite my team as well. So I think, it's there, right? And it's there to stay. And some people have to understand that, I think. Anyway, on the challenges, especially in surgery, we have a data absence problem, not just sparsity. So if you imagine a surgical team, what's going on? I mean, it's not just one person, it's a whole team operating, and they see with their eyes. So this is currently not recorded unless you do minimally invasive surgery. They feel, they do palpation for the tumor, for example. They listen to the sound of instruments interacting with tissue to do an assessment, and none of that is currently being recorded. So as a surgical data scientist, this can sometimes be tricky. And now with the OR black box, I think we have some potential to change this. This is a black box, courtesy, by the way, to Theodore Grancher from Stanford, that operates it similar to the black box in an airplane. So the idea is to have that box that captures everything so you can find out if something goes wrong, what went wrong. And I think that is a great idea. Now, again, the devil is in the details. So we're now basically, I would like to roll it out in Heidelberg, and the chief surgeon is also on the same page. But of course, the people in the ORs are like, but my dirty jokes when I'm operating, will they then be available to everyone, and all those issues, right? So what's the way out? I think one way out could be the patients, actually. So Theodore told me that when they rolled it out, patients started saying they want to be operated by the transparent doctor. And maybe if there's some more pressure by patients for such, let's say, systems, that will also trigger some change. And then finally, I mentioned it already, all sorts of biases, shifts. I think this is really, really a key issue. You also saw in the examples at the beginning, that's the environmental factors, degraded system performance, that's the key thing. And one that is very important, I think, in medicine is automation bias. So for example, if you imagine a colonoscopy for colon cancer screening, I mean, we all have to do it sooner or later, right? The way it works, they put the device in and then they retract it gradually and see if they find a tumor. Now, it has been shown already that, and I didn't find the publication, I'm not sure if it's already out, but that physicians are just waiting for the boxes to appear. So it's not our wish of complementing physicians' expertise with the AI, but now they're waiting for the boxes. And it may be more trivial to solve for, let's say, static images, because you can just disguise or not show what the AI says, although, of course, the problem that the physicians know the AI will look at it later. But in the intervention setting, it's even more tricky, right? Because what do? If you don't show the box at that moment, then the physician moves on, and then he or she should still look at it for second opinion, but then do you have to move the device back in? So all these things, automation, bias, how we integrate humans and machines, I think that's something that we have to give much more thought in. And yeah, now I'm actually exactly at 1 hour. Thank you so much for your attention, and of course to the collaborators in the team for allowing me to show all this content. Sure.
My question is more focused on eye. I'm not a computer scientist, I'm more of a business person. In your earlier slides, you mentioned about the deployment in ophthalmology is pretty low when compared with cardiology, etc. Remember in your earlier slides?
Yeah.
bar chart?
So you're talking about the system for diabetic eye disease that was put in practice in Thailand?
Right.
Yeah.
In general, how to use AI for detecting or diagnostic eye problem. So my question to you is, if the deployment is low, what are the major reasons? Would that because of more of the imaging technology, which gives bad data? You have this string of
yeah.
Is it because of the source, that is the data collection, or other reasons? And based on your knowledge, are you aware of any breakthrough technology going on, such as imaging, that might change the game, that will increase the deployment of AI in detecting eye disease or during helping the operations?
But not specific for eye, but for in general, you're asking, right?
Yeah. General eye disease, not just diabetic.
Okay. So the question is, what is the actual reason for deployment failure? I showed this example of eye disease. So I think in many cases, it's what we call distribution shifts. So it's the fact that we train the algorithm with images, for example, from a specific vendor, or that were taken in a specific manner, that then in practice, when any person operates the system, they don't maybe follow the protocol as closely. They do things a little bit differently, which results in the fact that mathematically, the images that serve as input for the neural network, they're not the same. So the question is, how can we deal with it? Well, I think ChatGPT shows quite well that if you have a lot of data, some of such effects actually do average out. I think that's one answer. And then there's also a lot of research going on. So for example, you can train algorithms. Let's say you have data from different devices, and you can train your algorithm in a way that it cannot recover which device was used to take the image. And that the hope is that through that, you become more robust to the specific modality that you use to take the images. So there are approaches, and also, I mean, I think we do see success stories also in eye. I think there was a DeepMind paper also out there. I can look it up for you. But in general, I think the domain shifts are the core problem, if you ask me.
Hi. As a clinician and a chair in informatics, I had a very long interest in diagnostics. One of the key things that we think about clinically is called spectrum bias. And what that means is that when you change your prevalence, it doesn't change according to Bayes' theorem with your sensitivity and specificity. There's a difference, there's a bias. And it arises because of things that you would not have otherwise predicted that differ between the settings, usually on how the marginal results fall one way or another. Very hard to predict. So the medical domain for a long time has settled on the view that you collect your data from the domain and the setting in which you're going to apply your diagnostic test. And you wouldn't use, for example, a balanced thing. You would use an imbalanced data set to do that, and you just generate a large sample. And it seems to be a real issue, actually. If you need a balanced data set to train, yet in order to get good validity, you really would ideally train on an imbalanced data set because that reflects your setting. How do you think that is going to change in the future?
Yeah, that's a tricky question. So I'm not sure if you heard the question, but it was, okay. One strategy to overcome issues with respect to prevalences is to just train an algorithm with the prevalences that you expect, right? So this is also a reasonable approach. I personally think, okay, there's some problems with it. I mean, as you know. So one is that if you have, typically you have very low prevalences of many diseases. And then everybody working on classification knows if you have an underrepresented class, then the performance will degrade drastically. So then if you train, so this is why oftentimes balanced training is done. The thing with the training in the setting that you would expect, I think it makes sense to some extent, but also prevalences do change, right? So my way out would be to train not with the prevalences you expect in case you have very rare classes, but to oversample rare classes, but to adjust the calibration to what you expect in practice. But that's my current view, maybe. Let's see.
Great talk. So I just have a question regarding the future of superintelligent machines in medical imaging. So how do you envision those? Like, will they be more personalized, which means they will be evaluated using their own subset of metrics? They will have, their own constraints in terms of, knowing if there is a prior data shift or imbalance or prevalence. So do you see the future more personalized to specific data, a subset of metrics, or is it more or is it the other side, building generalized models that can work on anything? So is it more so do you get my question? So
yeah.
Do we want to design more focused models to particular tasks, particular metrics, particular data sets? Or do we want to design a future AI that solves almost everything, like a universal model that generalizes across almost everything?
Yeah, so I think ChatGPT shows that a generalist model is kind of possible. I mean, it's just the first generations now, and I think it's showing, I was very surprised. I personally wouldn't have thought this to be possible, to be honest. So I think we will have foundation models as the basis. Now, the question is, how do we fine-tune them, and what do we do in the, I think they will be very integral part, but what do we do at the very end? This, I'm not sure, also because it has regulatory implications, right? So we will not be able to certify something like ChatGPT for the clinics because you need to show value for each, individual question that you're asking. So I think only because of regulatory issues, we will start with more targeted systems, but they will have a foundation model that they learn with. So it's basically hybrid to start with, and then let's see where we go.
Thanks for the great talk. It's very impressive with the level of skepticism. And my question is essentially how do you roll this kind of skepticism out to other labs which, for whatever reason, maybe they didn't know about what to be skeptical about, maybe it's easier to publish.
Yeah.
It's the less strong evaluations, maybe how so much of this is very deep in data. How do you actually make this the standard that the field holds itself?
Yeah.
Rather than the standard that you hold yourself.
Yeah, it's a good question. So how do you roll skepticism out? Because I think now we're back to the metrics, right? The metrics with which we are assessed in our field do not they do not encourage this way of thinking, right? Because they encourage you to publish a lot of papers very soon. So I was also not criticized by my team, but my team told me, you know, Lena, you put so much quality control on what we're doing that we always get much worse results compared to our competitors because they have leakage and they don't. For example, I have a very strong opinion on having a completely untouched test set. And this may sound easy at first sight, but sometimes it's not, because what we do is basically we do all the validation training as usual, then we have a test set in which we can, you know, overfit in the sense that we can try a few times for model selection, and then we say, okay, now we fix it, and we only do one test run. So then what do you do if this is not satisfactory, right? Do you do one more minor modification? And I think almost nobody does that, and they say, okay, but this is so hard. But then I told them, okay, if you think so, but then this would mean that our solutions are actually cutting edge. So let's start participating in challenges. And it turns out I think most of the times we won the challenges. And this gives them satisfaction, I think, because they see it actually makes sense to dive so deeply. But also, I think that now, so I've been doing this for a while, and I have had interesting reviews, for example, for our papers. I remember one Mikai paper in which we had 2 strong accepts and 1 strong reject. So we shouldn't suggest that the whole community is cheating. We never suggested it, but so I think it was very hard to publish such papers, but I think my perception is there's more room for it because people have become aware of, you know, the hype not reflecting what ends up in practice, and there must be some gap. So I do think there's more room for skepticism, but I think you're raising an important point. And what I haven't figured out myself is, I mean, how to publish negative results, because how can you be sure that your negative results are actually, that they generalize? I mean, having supervised PhD students, maybe one of them would have given me a negative result and another one a positive result. So if you have a positive result, it's easy to publish. If you have a negative result, did you really do things right to claim this is not working? I mean, so overall, I cannot give you a final answer, but it's a very good question.
Yeah, hi, thank you very much for your talk. I want to ask you whether you think that some of the problems you're talking about, in particular, for example, using accuracy, there are definitely communities in our community that understand completely that accuracy is a stupid idea and a stupid thing to think about with unbalanced data. It doesn't measure what you want to measure. On the other hand, it's absolutely clear that a vast number of people spend their entire time measuring success with accuracy. And I wonder whether these are really problems of sociology or science. I mean, we have groups out there that understand, doctors, for example, understand that you want diagnostic tests and you want confirmatory tests. You don't expect the one test to do everything. It's really about, I mean, it's certainly important, and certainly managing it and dealing with it and explaining it and teaching is absolutely key research. But is it science or is it social science?
You know, it's a good question. So I don't know if you heard it, but so accuracy is known to be a poor method for a poor metric for unbalanced data. So isn't it more like a social problem to deal with, or an education problem not to use that metric? Obvious, yeah. So okay, first of all, in our metrics reloaded framework, we don't really recommend accuracy. We only recommend it in the case the prevalences really reflect your target population, for example. So normally we don't use it or recommend it as a metric. But I think there's a different aspect to it. For example, if you train a classifier with the standard losses, cross entropy, for example, it so implicitly means you're optimizing for accuracy, right? Or argmax operator, which you use for converting your softmax scores into decisions, implicitly means you're optimizing accuracy. So I think accuracy as a metric, I fully agree with you, it's poor, but it has important links to other mathematical concepts that we use throughout the problem. So this is, I think, one problem. apart from this, I fully agree with you. When I talk to clinicians, they always want to report accuracy because it's so easy to understand, right? It's a fraction of correct versus everything, the fraction of correctly classified samples. But and then I suggest to them all these metrics that I think are mathematically much better suited, but I have to admit they're not as easy to interpret as accuracy. So yes, I agree. We also need to work on that other front, not just the mathematical part.
One of the problems is the anomalous results. So for instance, a patient who had multiple conditions may not be identified as having a particular condition because of the overlap. So where there is an expectation of a particular display, it may not be displayed because in some way the patient is anomalous. Is the artificial general intelligence more likely to be able to identify the blurred lines where there is a positive result but doesn't display in the way that is expected from a very specific set of identifiers?
Okay, the honest answer is I don't know, but I can share an anecdote with respect to ChatGPT because I just talked to a physician who deals with rare diseases all the time. And he shared with me an actual true story of a patient who went from doctor to doctor. He had a lot of symptoms. It was really unclear what was going on. And then he asked ChatGPT 3.5. And that gave a good answer, but was actually not the reason. And then he asked exactly the same question to the 4 version. And the 4 version came out with the right hypothesis. And the good news there was that the condition was actually treatable. So in the end, you could say ChatGPT in this case actually did help detect such a rare disease. So it seems to be possible even for rare cases in that generalized model with imaging, etc. I don't know, but I would be hopeful.
Okay.